Hello students, in today's lecture we are going to discuss the structure of the DNA, the structure of the tRNA as an adapter molecule and the some part of the protein synthesis, that is the translation part. So first we will see the tRNA as an adapter molecule. As an adapter tRNA as an adapter molecule or as an adapter. So tRNA, just in the previous year we have seen that there are three types of the RNAs which has the direct role in the protein synthesis. We'll revise some of the points of this RNA. That, as you know, there are three types of RNA. One is called as the mRNA. Some part of it we are already discussed in the 12th standard during the transcription. This mRNA stands for messenger RNA, and the role of this mRNA is that it always carries the message for the protein synthesis from DNA to the ribosome. Second type of the RNA is rRNA, which is called as the ribosomal RNA. This rRNA always attached to the ribosome, and when any mRNA molecule comes close to that ribosome, this rRNA gives the specific binding site to the mRNA. So role of the rRNA is that it provides the specific binding site to the mRNA. And the third type of the RNA is called as the tRNA, that is the transfer RNA. That means these RNA are involved in the transfer of the amino acid, which is the raw material for the protein synthesis, towards the ribosome. So these three are the types of the RNA. This theory is totally given in the 11th standard book, but here we are going to focus on only one type of RNA in detail, and that is called as the tRNA, because we have to see the structure of this tRNA. Generally, you should know that this tRNA is of two types. One is called as, there are two models of this tRNA. One model is called as the hairpin model, and another model is called as the clover leaf model. Okay, so there are two types of the models of this tRNA. One is like the pin, which is present in our hair, that is called as the hairpin model, and another model is called as the clover leaf model. So we will see the role of the tRNA as an adapter molecule by sketching the diagram of the clover leaf model of the tRNA. Okay, so first we will see why this tRNA is called as an adapter molecule by the diagram of this clover leaf model. So just I have told you that this tRNA has the role of transfer of the amino acid towards the ribosome during the process of protein synthesis. If we see the structure of this tRNA, then you will find that this tRNA has one short arm and one long arm. See here, the diagram that I am drawing here is very important, because this diagram is asked for the theory exam to sketch and label the structure of the tRNA. So you should know clearly the diagram of tRNA. This tRNA has two ends. One end is called as short end, that is called as the 5' and one end is called as long end, that is called as the 3'. Then this tRNA has the three arms. See here, these are the arms of the tRNA, and at the end of this arm there will be the loop formation. So here, this is one loop, this is another loop. Okay, these are the two ends of this tRNA. Now there will be another loop is there, and that loop is the important part of this tRNA, and that is called as the anticodon loop. See here, this is the structure of this tRNA molecule. We know that if there are base pair, then they will show complementary base pairing. So here we show that it is a base pairing. Important point is that here there is an attachment of the amino acid. So it is the amino acid. Here it is CGA. Okay, this is called as anticodon loop. These are actually the anticodon. See the structure of this tRNA molecule. What you will see here, that this tRNA molecule and the diagram of it is called as the clover leaf model. And here, if we draw the diagram of mRNA, then what will happen? C pairs with G, G pairs with C, A pairs with U. Such a type of U. These are the codons, and these are the anticodons. Now see here, it is the structure of the tRNA molecule. First we will see why this tRNA molecule is called as the adapter molecule. The meaning of it is that adapter means simply a bridge. If this tRNA molecule acts as a bridge in between two molecules, one is called as the amino acid and another one is called as the mRNA molecule. So in between this mRNA molecule and the amino acid, one bridge is there, and that bridge is nothing but the tRNA molecule. So adapter meaning the bridge which connects two different molecules. Thus this tRNA connects two different molecules, amino acid molecules and the amino acids and the mRNA molecule. And another meaning of this adapter molecule is that this tRNA adapts in such a way that it at the same time binds with the amino acid and at the same time it get joined to the codons of the mRNA. So this tRNA has the anticodon by which they get attached to the codon, or they get paired to the codon of the mRNA, and they get attached to the amino acid. So it is called as an adapter molecule. Okay, can you understand clearly? That is, this tRNA gets attached to the amino acid as well as it gets pairs to the codons of the mRNA. So it is called as an adapter molecule. Another meaning is that it acts as a bridge between two molecules. It connects the amino acid as well as it connects to the mRNA molecule by its anticodon. So it is called as the adapter. Now the structure of this tRNA, what it explains? It explains that this tRNA molecule has one short end that is called as a 5' end and one large end that is called as the 3' end. Okay, this 3' end of the tRNA has an unpaired sequence, and that sequence is called as CCA sequence. The sequence of the tRNA at 3' end has a sequence that is called as a CCA. This is very important because this sequence is present at the 3' end of the tRNA, which is unpaired. It do not pair to any other base pair, or it do not show complementary base pairing to other strands of the tRNA, because here it is a large one and this tRNA N has do not any complementary strand opposite to it, because opposite to it is a 5' end and it is very small one. So this CCA sequence is called as unpaired sequence. It do not pair with other strand of the RNA, and that's why it is called as an unpaired sequence of the tRNA. So at the 3' end, at the 3' end of the tRNA, there is a unpaired sequence that is called as CCA sequence, and at that sequence there is the attachment of the amino acid. So here you can see the amino acids are always attached to the 3' end of the tRNA, while at 5' end there is no any attachment of amino acid, and you should know that at 5' end there should be one base pair, and that base pair is called as the guanine. Okay, in our textbook the base pair is not given, but you should know that at the 5' end there is present the guanine base pair, and at 3' end there is present CCA base sequence, which is called as a unpaired base sequence. And at which end of the tRNA this amino acid get attached? This amino acid get attached at the 3' end of tRNA. Then we have seen that there are 3 arms. These are the arms. Okay, this small structure is also called as the short arm, or it is also called as the lump. Okay, these are the 3 arms. They has one of the short arm, which is also called as a lump, and at the end of this arm there are called as the loop present. These are the loops present. But in this structure we have to concentrate only on one loop, and that loop is called as an anticodon loop. This is the anticodon loop. Just in the previous lecture we have seen that these anticodons are always present on the tRNA and codons are always present on the mRNA, and there is always complementary base pairing between this codon and the anticodon. So the loop which is present on this tRNA, which we call as an anticodon loop, and to this anticodon loop there are 3 bases are there, and these are, here we have written CGA. These are the 3 bases which we call as a anticodon, and this CGA base pair is complementary to the codon on the mRNA, and codons are C pairs with G, G pairs with C, A pairs with U. So there is always codon and anticodon pairing found between this tRNA and RNA. That means in the cytoplasm near the ribosome, this tRNA is always available with the amino acid which is attached at the 3' end, and when it comes close to the mRNA molecule, then according to which pair of anticodon present on that tRNA, that anticodon always get attached to the complementary codon on this mRNA, and thus there will be codon anticodon pairing will be found in this tRNA molecule. So actual structure of this tRNA is like the inverted L shape. The structure of the tRNA is like inverted L shape. L हा लेटर जर आपण उलट करून बघितलं तर जसं स्ट्रक्चर पाहायला मिळतं, तसं जवळपास आपल्याला स्ट्रक्चर हे tRNA मॉलिक्युल्सचं पाहायला मिळतं. सो दिस इज द इम्पॉर्टंट part of the tRNA as an adapter molecule. Very small simple questions will be asked for the MCQ point of view, and those questions are that which is the end of the tRNA to which the amino acid get attached, then that end is the 3' end. Second question is at which end of the tRNA the unpaired sequence ... is present, that is the CCA sequence, cytosine and the adenine. Then which is the short end of the tRNA, that is the 5' end. At the 5' end which base pair is present, and the answer of that is it is the guanine base pair. It is the guanine base pair. Now then another question is that how many loops are there in the tRNA? In the tRNA overall there are 3 loops, and the loop where the anticodons are present, that is called as the anticodon loops. These anticodon loops are complementary to the codon of the mRNA, and the structure which is shown here, that is nothing but the clover leaf model, which actually appears the inverted L shape. Okay, so few questions are very important from the structure of this tRNA molecule. For MCQ point of view, also you should know the diagram of this tRNA molecule, that is which end is a short, which end is a long, how many loops are there, how many arms are there, which is the lump or the short arm, where the anticodons are present. This information is very important point. Okay, now we will see the second part of this protein synthesis after transcription, that is the translation, because in the starting of the protein synthesis we have shown that the central dogma that states that the DNA from the mRNA, that process we have called as a transcription, and this mRNA from the protein, that process is called as a translation. But after completion of the transcription, we have seen the what is meant by genetic code. And after completion of the genetic code, here we have studied the structure of tRNA first, and then now we have to study the translation, because for getting the information from the translation point, we should know what is tRNA, where the amino acids get attached to this tRNA molecule, where the anticodons are present on that tRNA molecule, what is meant by genetic code, how the mRNA transform is how the DNA transforms its codon on the mRNA, and then mRNA carries that codon towards the ribosome. For getting such information, after getting this information, you will get the idea of the translation. So translation is the second important part of the protein synthesis that we are now going to discuss here. So again, see here, this is a diagram of the tRNA molecule, clover leaf model. If it is asked for the theory to draw the diagram, then you should draw this diagram of the tRNA molecule. So 2 points are there. For theory, the diagram is asked, and for the NEET exam, the MCQ that I have revised here are very important one. Now we will see the next point, that is called as the translation. Now we will see first the definition of the translation. What is mean by translation? Now here we have to see that codons or mRNA have the role in the process of translation. So what is mean by translation? Translation is the process in which the codons on mRNA. Translation is the process in which codons on mRNA are get translated. Codons on mRNA are get translated, and here the specific amino acid sequence in a polypeptide chain will be formed on the ribosomes. Codons on mRNA are get translated, and a polypeptide chain is formed on ribosome having specific sequence of amino acids. Here, okay, what is the definition is given here? Codons on the mRNA. No doubt, codons are present on the mRNA. These codons on mRNA are get translated, and when this codon get translated, a polypeptide chain is formed on the ribosome. Why the polypeptide chain is formed on the ribosome? Polypeptide chain is nothing but the proteins. Why they are formed on the ribosome? Because ribosome is the site of protein synthesis. Ribosome is a protein machinery. So on that ribosome there will be formation of the polypeptide chain, and that polypeptide chain only form when this codon get translated. And these ribosomes which has the polypeptide chain, this polypeptide chain have the specific sequence of the amino acid. The sequence of the amino acid is in a specific manner, because in a genetic code lecture we have seen that in any polypeptide chain the first amino acid is methionine. So that polypeptide chain also has the sequence of amino acid, and that sequence should be in a specific manner. That means in that polypeptide chain there should be the first amino acid which codes for the methionine. Okay, so we have called it as a specific sequence of amino acid. This methionine should be first in the polypeptide chain. It should not be in the middle or at the last of the polypeptide chain. So it is very important that the specific sequence of amino acid should be there in a polypeptide chain. Then what is mean by the translation? Here one word is given that codons on mRNA get translated. Get translated means just in the tRNA structure we have seen that when this codon on mRNA get paired with the anticodon of that tRNA, then that process is completed, and here particular amino acid will be linked on the ribosome. Suppose if here this is the mRNA and it has a codon AUG, okay, then what happens to this AUG codon? The anticodon will be UAC. So if this is complementary pair occur, and then there will be one amino acid was linked, one amino acid is linked on that ribosome, and that amino acid should be methionine. Okay, so when codon and anticodon complementary pairing takes place, there will be amino acid formation. But which amino acid formation? According to the sequence of the codon on the mRNA that we have studied in detail in the genetic code. So here that amino acid will be decided by the codon on the mRNA. But for the decision of that codon on the mRNA, there should be complementary base pairing to the anticodon. Then and then only there will be one amino acid formation on that ribosome, and that is called as the translation. So very clearly observe the definition of the translation, that translation means the codons on mRNA are get translated and a polypeptide chain is formed on the ribosome having specific sequence of amino acid. This is about the translation. Now we will see what is the important point is there, which factors are essential for this translation. So this translation process occurs on the ribosome, because the ribosome is the protein factory where these proteins are formed, where the translation process occurs. All the proteins which are nearly about the 20 amino acids which are involved in this formation of the proteins, these proteins are formed inside the cell. So that process is called as a intracellular. Not these proteins are formed outside the cell. Why I am telling this? Because you know that the transcription process where the mRNA formation forms from the DNA are occur in the nucleus part of the cell. Okay, and the translation process where mRNA forms the protein, it occurs in the cytoplasm of the cell, because in the cytoplasm there are the ribosomes. So mRNA, after formation from the DNA in the nucleus, it migrates to the cytoplasm where the ribosomes are present, because this mRNA has to bind to that ribosome. So here you will see that all these parts are of the cell and all the proteins are occur inside the cell. So that process is called as intracellular. They are not found outside the cell. It is not an extracellular process. Now we will see which requirements are essential for the translation process. This translation process includes important factors, and those are, there should be the first requirement is amino acids. Amino acids are there. Then the mRNA molecule should be there. Then tRNA molecule. Next one is ribosome. Then ATP. ATP is also there. Then one of the important ions which are responsible for binding of larger and smaller subunit of ribosome is the Mg double plus ion. Different enzymes are there which are required for activation of this amino acid, activation of the tRNA molecule. So different enzymes are important. Then there will be some elongation factors are there, some initiation factors are there, that is important. So elongation, then translocation, and third one is releasing factor. Observe carefully what we have written on the board. See here, these are the basic requirements for the process of the translation. Amino acids, mRNA, tRNA, ribosomes, ATP molecule, Mg double plus ion, different enzymes, elongation factor, translocation, releasing factor. Now we will discuss some of this point, that is amino acids are the basic requirements for the protein synthesis because it acts as a building blocks or the raw material of the protein synthesis. So it should be required for the translation. mRNA molecule is required because mRNA has a codon and that codon decides which is the sequence of the amino acid in a polypeptide chain, or that codon particularly code a particular amino acid. So mRNA is important. Why tRNA is essential? Because this tRNA transfer the activated amino acid, or this tRNA carries at its 3' end the activated amino acid and it transfer it towards the ribosome when there is a codon-anticodon pairing takes place. Then the ribosome is a cell organelle which is present in the cytoplasm and it acts as a protein synthetic machinery. On that ribosome actually the polypeptide chain gets synthesized. So it is very important. This ATP means adenosine triphosphate, which acts as an energy source because for all translation process energy is required and that energy is given in the form of ATP molecule. Mg double plus ion, that means as we know that ribosome has two subunits, one is smaller subunit and one other is larger subunit. So these two subunits are always not get attached to each other. They are free in the cytoplasm of the cell, and when the translation process occurs, these two subunits, small and large subunit of ribosome, should get attached to each other, and for their attachment there will be requirement of one ion and that is called as Mg double plus ion. Then enzymes, because the amino acid which is acts as a raw material which is present in the cytoplasm, these amino acids before process of the translation is in inactivated form. So that amino acid should be converted into the activated form, and for activation of the amino acid as well as for activation of the tRNA molecule there will be requirement of some enzyme like amino alkyl synthesis enzyme or peptide transferase enzyme for the polypeptide chain synthesis. So different enzymes are there, we will discuss it later. So these enzymes are essential for activation of different molecules which are required for the translation process. Then what is meant by elongation? Elongation means increase in length. See here, first amino acid will be the methionine, then there will be formation of second amino acid, then third, then fourth, then fifth. Such amino acids are linked to each other and there will be long elongated chain of the polypeptide. So attachment of this different amino acid by the peptide bond towards each other or linking of this amino acid is possible only due to elongation factor. So elongation factor should be there which elongate this amino acid chain. Okay, so it is an elongation point. Then translocation. Translocation means what? Here this is the mRNA molecule. On this mRNA molecule at the first side there will be attachment of this ribosome, and the first site is the 5 prime end, and the end of this mRNA is always we have seen that 3 prime end. So at the initiation site of or at the start point of the translation, at the starting of the translation, this ribosome is present at the start codon, where it is always initiation codon is AUG codon. In characteristics, we have seen that it is the AUG codon which is present at the start of the 5 prime end. So that codon should be matched with the anticodon of the tRNA. So here first this ribosome attached towards the 5 prime end where initiation codon is present, AUG, then it migrates towards here, then here, and at the end it gets migrate to the last part that is called as the 3 prime end. So first position of the ribosome is at 5 prime end, then it moves slowly and then finally it attaches to the 3 prime end. That movement of the ribosome on the mRNA strand is called as the translocation. So translocation factor should be there, due to which these ribosomes move from start to the end of the mRNA molecule. And last one is called as the releasing factor. After completion of the polypeptide chain, that polypeptide chain should be released from this mRNA. So suppose this is the ribosome, and on this ribosome the polypeptide chain will be formed. So this polypeptide chain should be separated from this ribosomal unit, and that requires some releasing factors, and these releasing factors are present here, and by which this chain of the polypeptide chain can be released from the mRNA molecule. So I think you will get clear idea about this amino acids, mRNA, tRNA, ribosome, ATP, MG double plus ion, enzymes, elongation factor, translocation factor, and the releasing factor, which are the essential components of this translation process. Okay, so this is about the requirement of the translation by which the synthesis of the proteins takes place. Next to it, we will discuss some of the important points, and that points are the important factors before the process of translation that we are going to discuss. And in this, the first point that we are going to discuss is the requirement. In detail we are going to discuss here, that is there should be requirement of 20 amino acids. As we told that these 20 amino acids are present in the cytoplasm, they are present in inactive form. So these 20 amino acids should be get activated first, and that activated amino acid then they are present in the cytoplasm, and later on they form the polypeptide chain. So first requirement, that where these amino acids are present, how many amino acids will be required for the translation process. These amino acids are present in the cytoplasm. There is a requirement of the 20 amino acids, because in the genetic code we have seen how many amino acids are involved in the synthesis of protein. Some of the names we have given, that is the tyrosine, methionine, leucine, valine, glutamine, glutamic acid, aspartame, aspartic acid. So different phenylalanine, different amino acids are there, and that should be 20, and it should be present in the cytoplasm, and that forms the polypeptide chain. So it is the first requirement. Then second requirement is given there, that is for the translation process or for the whole protein synthesis, main important part is the DNA. So DNA controls protein synthesis. DNA controls the protein synthesis. How this DNA control protein synthesis, that we have seen number of times, but some students cannot understand the meaning of this line, that DNA controls the protein synthesis. That means first this DNA form the process of transcription, where you will find that on that DNA codons are get transferred on the mRNA, and from mRNA proteins are formed, and that process we have called as a translation, because these two terms we have seen in the central dogma. So transcription and the translation, these are the 2 points by which this DNA controls the protein synthesis. So main important thing is that who controls this whole protein synthesis, that means the DNA molecule by the 2 process, transcription and translation. So this second point is also important, that how DNA controls the protein synthesis. Then the third important part is there, that the RNA molecules, this RNA molecules acts as an intermediate molecule. These RNA molecules acts as an intermediate molecule in protein synthesis. What is meant by intermediate molecule? RNA acts as intermediate molecule between the DNA and the ribosomes. Between the DNA and the ribosome, because DNA has information for the protein synthesis. A ribosome is the cell organelle where actual protein synthesis is formed, and in between these 2 important molecules there is another molecule that is called as the RNA molecule, which form the intermediate molecule, because they are present in between the DNA and the ribosome. And we have seen that 3 types of RNA has equal role in the protein synthesis: mRNA, rRNA and the tRNA. mRNA carries a message for the protein synthesis towards the ribosome. rRNA always attached to the ribosome, and when mRNA comes towards the ribosome, this rRNA provides binding site to the mRNA. And tRNA, we call it as a transfer RNA. It transfers the activated amino acid to the ribosome. So this mRNA is also important, but it is important and it acts as a middle role between the protein and the ribosome. Then the 4th factor that is given, and it is that the ribosomes has the important role. That means it acts as the unit for the protein synthesis. It acts as a protein factory where their proteins are formed. As we told that this ribosome has the small unit and the large unit. So if we see the structure of this ribosome, you will find that such a structure will be there. It is a small unit, it is a large unit. See here, these both units, small and large, are not always attached to each other. They are always free in the cytoplasm of the cell. But when there will be formation of the proteins, these 2 subunits join together, and the joining of these 2 subunits occur with the help of one ion that is called as Mg++ ion. So Mg++ ion has a role in joining of these 2 subunits, smaller and larger subunits, during the synthesis of the protein. Otherwise, when protein synthesis is not taking place, these 2 subunits are always free from each other and they are present in the cytoplasm of the cell. So this is about the 4th point that we have seen about this ribosome. Now the important point is here that this ribosome, which we have called as a protein machinery, this ribosome has 3 important sites that now we are going to discuss here. So these are the similar points that you are well known about it, but line to line expression of the textbook are discussing here, and that's why I have given the point that amino acid is required, DNA control protein synthesis, RNA acts as intermediate molecule between the DNA and the ribosomes, and the ribosomes is the protein machinery. Now here we will see which different sites are found on the structure of this ribosome. So if we see, this is, suppose this is the ribosome. Okay, this structure is of the ribosome. Here we will label it as a ribosome. This ribosome has 4 binding sites. One site of this ribosome binds to the mRNA. So this is the mRNA molecule, and to this mRNA, one side of this ribosome get bind, and other than it, there are 3 sites, and those are here P site, A site, and here is the E site. What is it? A site P side and E side. Okay, A side means we call it as a arrival site. P side means it is the polypeptide site. And E site means it is exit site. Now, see here, what is meant by it? Now, you have to see that peptide tRNA site, amino acid tRNA site, and last one is the exit site. So here the A site means arrival site. For our understanding, the meaning is given that arrival site. What happen on these ribosomes? The tRNA get attached. Those tRNA which has at their 3' end reactivated amino acid. So here at the A site, amino acyl tRNA site get attached. What is meant by amino acyl tRNA site? So if we see, this is the structure of the hairpin model, then at the 3' end of that tRNA molecule there will be amino acid. But that that tRNA molecule get charged by one enzyme, and the name of that enzyme is amino acyl synthetase. What I am saying here, this is tRNA and their 3' end there is amino acid get attached, and that tRNA get charged, and that charging of the tRNA, that tRNA gets activated by enzyme, and the name of that enzyme is amino acyl tRNA synthetase enzyme. So the enzyme's name is given to that tRNA, and the tRNA is called as amino acyl tRNA. That tRNA is called as amino acyl tRNA, and that amino acyl tRNA is ready or get attached to this A site, and so according to the amino acyl, the name is given as the A site, because A site is that site where the tRNA which is activated by this amino acyl synthesis enzyme, and the name of that tRNA is amino acyl tRNA, get attached to this A site. So A site means amino acyl tRNA site, or it is also called as a arrival site, or that is a start point on the ribosome where tRNA get attached. So arrival or the amino acyl site. After that there will be P site, and the P site is given by two names. That is, here actual polypeptide chain is synthesized on the ribosome, or it is the site where you will find the peptidyl tRNA site. That means here after the A site, this tRNA goes to here, and this tRNA gives its amino acid here, and by the complementary pairing of the codon and anticodon, there will be formed first amino acid, then second, then third, then fourth. Thus a polypeptide chain is actually synthesized on that P site, so it is also called as a polypeptide site, or the tRNA peptidyl site is there, because this tRNA migrate to here, and that tRNA is given name as a peptidyl tRNA because they use their amino acid here, and a long polypeptide chain will be formed here. So it is a peptidyl site. And the last is the exit site. So here after giving its amino acid by tRNA to ribosome, then that tRNA is free, and that tRNA comes here, which is the empty one, which do not have any amino acid at its 3' end, and that empty tRNA goes from this site to outside the cell, and that is called as the exit site. So here what we have seen, that we have seen the three sites of the tRNA, that is the A site, P site and the E site. Okay, now the some part of it we will discuss in the next lecture. Thank you.